Right, shall we? Okay, number one, this is unit 2 paper, and uh, we could do only few papers before the exam because first paper is starting May, 6th of May. And I encourage if you could come physically to the classes, more effective than online. So effectiveness is high with the physical class than online class. And the first thing is, if you sit and you're waiting for the exam in August and you come up with the low marks, that is bad for everyone. Me, you, parents, everyone. So it is very important that we need all the strategies to increase the marks. It is not just the theory and we know biology, we write something, we get the marks. Concept is cannot work with Edexcel because Edexcel is highly unpredictable. There are paper patterns and no one can predict the paper patterns. You will mostly face the challenge to get the marks. Okay, so unit 2 paper. Now last few questions, that is 5, 6, 7, 8. The total marks is 43. And question number 1, question number 2, question number 3, question number 4, total marks 37, right? 37. Now the challenge for most of the students, many of the students, unless they are very extremely bright student, they can manage. Majority of the student ended up, they try to get maximum marks for the first 4 questions. So when they try to get maximum mark for the first 4 questions, we cannot guarantee they get a 37 out of 37. So this is not always 37 out of 37. Then there is another issue comes, especially for these questions, time management. Most of the students, they are not writing at least 1 or 2 questions of this category. 5, 6, 7, 8, they are not writing 1 or 2 questions because no time. Reason is they don't have time. Second thing, they are difficult. So as a result, out of 43, you get a lower marks. So most of the time, ended up in 10 marks. And here, if you get 20, if you get at least 23 around, your total marks is 33. Now this is the truth. So your grade is mostly C. And very tricky, if you go down, it's very close to D. C and D, there's not much different. They're very close. So target is about 40. So these are the challenges you face in these papers. So you are facing challenges for these papers. Now my strategy is first 4 questions you try to manage because it's easy, right? It's not that difficult to manage. Now let's go for question number 5. Start from question number 5 to 8. And if time left, we go to 1, 2, 3. So question number 5. A new type of bacteria was discovered in Greenland, and they're very old. The question is, describe how scientists could determine that this was a new type of bacteria. Now, how scientists decide this new type of bacteria? If you want to get 2 marks, so what is your exact points to get 2 marks? Right. Uh, in classification, scientists classify organism based on a morphology. Appearance. This was the first classification. The first classification is based on the morphology. And the second classification, what is second classification? Second classification based on molecular phylogeny. Then we identify new species based on these two criteria. This is the one still we are using. This is the first one, oldest one. This is the new one. Based on criteria of first and second one, we can identify the species. Then you write this bacteria can identify as a new bacteria by looking at the morphology, cellular appearance, and different structures present in the cells. And this is compared with the other bacteria. Second one, this bacteria can be classified as a new organism based on molecular phylogeny. Molecular levels, we are comparing with other bacteria such as DNA, RNA, and proteins, analyzed and compared with the other bacteria. So you have to write 2 sentences under these 2 categories. So did you write answer? No. Yeah, I think this kind of a question is very hard to get a correct answer. I mean, not only you, but most of them face difficulty when you see how we can see this as a new species. Then you have so much of information. I mean, you can write, you can write something, but you don't know how accurate is the answer, because it's a so much of thing you can write, but exact thing how scientists classify as a new organism based on this classification system. So classification system based on one appearance, similarities and differences. Other one is molecular level differences and similarities. So that's why from question number 5, things are getting very hard to get the marks. Okay, next question. we are not working with bacteria in AS level. We just only few things we just only learning few things: growth, survival by changing the conditions, the effect of temperature, and desiccation, that means the water level, on different bacteria was investigated. Graph 1 shows the effect of temperature on the growth rate of bacteria A and bacteria B. So this bacteria survive in a cold temperature and its optimum temperature is 10. This bacteria B survive in a higher temperature. Its optimum temperature is 90, around 90 to 93 around. It shows the highest growth rate at these temperatures. Graph 2 shows the percentage of survival of bacteria C and the mutated bacteria C after water was removed from the surrounding. So there's another bacteria. This bacteria is C, and they have mutated by removing the water. Percentage survival, bacteria C, mutated bacteria is less. After mutation, the survival is less. Explain the changes in growth rate and percentage survival of these bacteria. Use information in the graph and your own knowledge to support your answer. Okay, anyway, 6-mark question. We need a very good answer with a lot of timing, information in the graph, and your own knowledge. So in this case, you have one strategy. All your skills used to analyze the result, because you have a two result. So you use your all the result given in this graph to analyze. For example, when you start this graph, the growth rate of bacteria A increase from -5 centigrade is a low, it's a no growth. But when temperature increase, the bacteria growth rate increase, and the optimum temperature 10 Celsius degree, they have a highest growth rate. After that, bacteria A decrease growth rate, and temperature 20 Celsius degree, there is no growth of the bacteria. Same thing you can explain to bacteria B can grow in higher temperature. So there's no growth at 66 around, but the growth rate increase after 66 around, and then it goes to a highest rate at 94 around. Then that's the optimum growth rate, the bacteria at 94, and then decrease after 105 around. So if you completely describe this graph, your own knowledge, now how do you explain the effect of temperature on the growth rate of bacteria? So now you can explain why the temperature affect the growth rate of bacteria. For that, you can take different bacteria adapted for different temperatures. Bacteria A adapted for more cold temperature, bacteria B adapted for more high temperatures. So then you can write the basic theories. For example, the cold temperature, the kinetic energy is lower. So when the temperature is lower, kinetic energy is lower. As a result, enzyme collision is lower, and active site binding with substrate is lower, and the rate of growth is lower. But when it comes to the optimum temperature, the collision is highest, and the active sites are occupied, and more substrate are formed. Growth rate is higher. But bacteria B has different temperatures. They are adapted to different temperature, and their optimum temperature is 94. In that case, enzymes activity is higher at 94. So you use your enzyme theory to explain this graph. Then graph 2, they use a different bacteria C. Okay, water percentage of survival days after water was removed. So bacteria C reduced percentage of survival after water was removed. That means the graph described, so it was 100% from the beginning, but then it reduced to this much percentage after water was removed, like 60% reduced, up to 60%, reduced to 60%. The mutated bacteria, when the water is removed, their percentage survival decreased to zero level after 6 days. Hmm? Six, 9, right? So we can take 9 days. It's also there. It will be 9 days. And you can further analyze using this standard deviation, because both have standard deviations, error bars. And you can say, comparing the two bacteria, the standard deviation is not overlapping. The results are significant. Results are significant. Standard deviation is not overlapping. And then you can explain what is the role of water in a bacterial cell. So bacteria use water for their metabolic functions. If there is no water, they cannot do metabolic functions. Then mutated bacteria cannot adapt it for the surrounding changes, and the mutation cause them to lost completely after 9 days. So one important thing with the 6 marks question, when you analyze and you write your answer with at least 1.5 page, you consume a lot of time. So you have to really manage your time when you're writing the 6 marks questions, because you need a lot of analysis, and after that you have to write the answer. Sixth question. The antimicrobial, that the killing ability of bacteria of an extract from the plant was investigated. 3 sets of agar plates were poured. Each set had a different type of bacteria over the surface of the agar. Discs containing different concentration of this extract were placed onto the each plate as shown in the diagram. Now what happened here, if we have a bacteria and these are the different concentration use 0 as a control, 25 microgram and 50 microgram. So if we think this plant has antimicrobial properties, we take the extract and then we soak this disc with the extract. These are the paper disc. If you keep this disc on the agar plate, they are diffused to the agar plate. When they diffused, the highest concentration is here, and then concentration getting lower, 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 and then no. That's how the chemical diffused. Here is a higher concentration, and then concentration decrease, and then finally at certain point there's no chemicals. So it creates a ring around the disc. In this ring, closer to disc, the concentration is high. Away from disc, concentration is low. So that's a distribution of the antimicrobial extract from the plant extract. Now when it comes to bacteria here, bacteria has no chemicals, bacteria can grow. So bacteria is multiplied. So when it comes to this place, let's say like this concentration are like this. When bacteria comes to this place, there's a little effect on bacteria because concentration is here. They can still multiply. And when it comes to here, they can still have some effect like this. The effect is because when it comes to closer, concentration is high, they can't grow. So maybe they can grow somewhere only this area. And some can grow only this area. As a result, we can see the growth, different growth, because the white zone is no bacteria. If you use less concentration, the diameter radius is lower. But if we use higher concentration, they can grow and we have a big clear area. The diameter of each zone of inhibition was measured. The table showed the result of this investigation. Comment on the antimicrobial properties of this extract. Now, comment is a little bit similar to compare and contrast plus describe. So it is not completely compare and contrast. It is not completely describe. So you have to comment of anything which you see similar or different in each group. So extract concentration increase, mean diameter of zone of inhibition, bacteria A increase, bacteria B increase, bacteria C increase. Okay, from this way, 50 concentration is effective. So ABC bacteria all show the large inhibition zone at 25 concentration, again similar, and this is the control. There is no zone of inhibition. Zero means no zone of inhibition. So basically, we can say when the concentration increase, zone of inhibition is higher. So that means there is an effect. There's an effect of this extract that we should also consider that one. If there's no effect, then there's no zone of inhibition. But we see effect. When the concentration increases, there is a higher zone of inhibition. So this has antimicrobial properties. So all the three bacteria shows a similar result. So all ABC3 bacteria showed similar result, and at least we can see bacteria A is at least 14, the highest inhibition zone displayed by the bacteria A. So bacteria A is more effective to these antimicrobial chemicals. I think B and C are more or less same, right? Only A is more effective in both concentrations. If we have a pain, we get the drugs like a paracetamol. They are pain-killing drugs. Drug A has developed from another species of plant. This drug has been tested as a treatment for headaches. The effect of this drug on the duration of headaches in volunteers was compared with the drug B. So if we have done this, what is the case of the drug? Phase two. Yeah, headache in volunteers, right? So headache mean the patients. We can consider them as the patients. Was compared with drug B standard treatment for headaches. Drug B was standard treatment for headaches. Here is a drug B that is a standard treatment. So they compare with the standard treatment. So usually we can't take control. We take control as a existing drug. If there's a known drug, we use as a comparison. Right. Evaluate the result of this investigation. Now we are more interesting on drug A because drug B is the standard one because it is in the market maybe. But we are interested in drug A. So drug A reduces mean duration of headache. Overall, when the concentration increases, mean duration of headache decreases, right? Overall. Okay, first thing you can write, when concentration of drug increases, mean duration of headache decreases. And you can make a special point now to give the drug A is much better than drug B from 200 to 400 concentration. It shows higher reduction than drug B. So concentration 200 to 400, higher reduction than drug B. Higher reduction than drug B. But there is a continuation of this. You can still add something onto that. There is a reduction, but hmm? Now here there is a higher reduction, right? So here higher reduction, 2 and 4, compared with the drug B. Yeah, the error bars overlapping, right? Error bars overlap. This reduction is not significant. So this reduction is not significant. So here reduce with concentration. It is not significant. Error bars are overlapping. It is not significant. And we don't have a data of drug B from 0 to 200. So here we can say there is no data to compare from 0 to 200 of drug B. So there is no data to compare from 0 to 200 for drug B. I mean, we can't say this is better than drug B because standard deviation is overlapping, right? Drug A was then used a three-phase drug trial because we highlighted this is phase 2 trial. Compare and contrast phase 2 and 3 in contemporary drug trial using drug A. Compare and contrast phase 2 and 3. Compare and contrast phase 2 and phase 3 in contemporary drug trial using drug A. So similarities, both phase 2 and phase 3 used patients, yeah. So phase 2 and phase 3 used patients. I think we go for more differences. Because anyway, you get a mark and 2 marks you need for this one. So what are the differences? Phase 2 use less patients than phase 3. Phase 3 use placebo and double-blind study, whereas phase 2, no. I mean, we don't want to spend time. Because time is also precious in the exam. If you know clearly these are the clear point, just only write that one. Otherwise, we can write few things, but it's wasting time again. So here, you know that there are the clear differences and similarities of phase 2 and phase 3 trials. So question number 7. The photograph shows different varieties of eggplant roots. And these are the mass imported in by countries in tons: Austria, France, Italy, and Slovenia. It is expected that the import of eggplant by France would increase by 1.6 per year. So every year, constantly increase 1.6. Calculate the expected mass of eggplant by France in 2023. So after 3 years, what is the rate, what is the amount increase? So this is the 2020, and if you want to find a 2021, so 1.6% increase. So we have to get the amount which is going to increase, right? So what is the value for this? If you calculate, $846. This much 46. Okay, 0.2. Now this much need excess in the next 2021. So then you have to add this to 28920. Then the exact value is 28. Correct? Now what you're going to do, 2020, this is for 2021. Now you're going to add for 2022, but you can't do this. You can't do this. Why? What if they ask 2029? So you are wasting maybe 15, 20 minutes just to get a 2 marks. It's not worth. It's not worth for this paper, right? We need a method. We need a method. We need a method, but I'm pretty sure the learner syllabus exam, Edexcel's or Cambridge student, they don't use in IGCSE level this kind of a maths. Local syllabus student, they do this for O level by adding 1 year, 1 year, 1 year like that, right? So this kind of a maths is called compound annual growth rate. So we need to learn what is compound annual growth rate. Unfortunately, it is not in the textbook, but I have included in the notes, my notes workbook, 2021, similar question was asked. 2021 year, similar question was asked. The island of Santa Cruz, the population growth or something like that. As a result, we learned from the question papers. In maths, they included this compound annual growth rate calculation. So we have to learn this now, compound annual growth rate calculation. Now, compound annual growth rate calculation, we do like this. So this is the mass imported in tons. So that's a total, right? That's a total in the year. So if it is a total in the year, then this total should be multiplied by 1 plus 100, the growth rate. The rate is 1.6. This is the R, this is the T, and power for this how many years? This how many years? So Y is 3 years. Now this formula is not given in the exam. You need to remember this formula. This formula you need to remember how to calculate. So then this should be 1.6 divided by 100 is, this is 0.016. 1.6 divided by 100 is 0.16. 0.16 plus 1 is 1.016. And how many years? 3. And multiply by 52,892. So this you need to use calculator now. 1.016 power of 3 multiplied by 52,892. So what is the answer? 55,472. This mathematics is important because we don't get often in the text papers and the question. Rarely it is coming with these exams. Only